Welcome to the NIH Director's Wednesday Afternoon Lecture Series. I'm Julie Segre. I'm a senior investigator in the National Human Genome Research Institute here at NIH. Today's lecture is a special event in our WALL series, the annual Rolla Dyer Lecture. And this is the oldest continuous lecture at the NIH, established in 1950 in honor of the former NIH Director, Dr. Rolla Dyer, a noted authority on infectious diseases. And among his many accomplishments, Dr. Dyer is known for identifying the cause of endemic typhus and developing a vaccine to protect against it. Sounds pretty important these days, right? So Dr. Dyer was NIH's director from 1942 to 1950, a time of rapid growth for the NIH. He oversaw the creation of the Grants Division, the planning of the NIH Clinical Center, and the establishment of 3 additional institutions, heart, dental, and mental health. The Dyer Lecture features internationally renowned researchers nominated by NIH staff who've contributed substantially to medical as well as biological knowledge of infectious diseases. And what better speaker for the Dyer Lecture than Dr. Paul Turner, the Rachel Carson Professor of Ecology and Evolutionary Biology at Yale University School of Medicine. Dr. Turner is an expert on the evolution of infectious disease and antibiotic resistance, host parasite and vector virus interactions, and virus applications via phage therapy. Dr. Turner received his undergraduate degree from the University of Rochester in biological sciences, followed by a PhD in zoology from Michigan State University. His postdoctoral training includes positions at the University of Maryland College Park, at the University of Valencia in Spain as a NATO postdoctoral fellow, and right here on the NIH in the NIAID lab of clinical investigator Jeff Cohen. His research career has since been at Yale, where he arrived as an assistant professor in 2001 and became a full professor in 2011. At Yale University, Dr. Turner studies the evolutionary genetics and genomics of microbes, especially the ability of viruses to adapt or not to changes in their biotic and abiotic environments. These studies concern environmental challenges faced by viruses at all levels of biological organization, including effects of changes in molecules, proteins, cells, populations, communities, and ecosystems. Aside from scoring that NATO gig in Spain, Dr. Turner has received many honors. These include being named as one of Cell Press's 1,000 Inspiring Black Scientists in America last year, election to the National Academy of Sciences and to the American Academy of Arts and Sciences, a recipient of the Innovator Award from the Blavatnik Fund, and the invitation to deliver a multitude of named lectures such as this Dyer Lecture. I had the chance to work with Paul as a member of the National Academies of Science, Engineering, and Medicine Committee on Data Needs to monitor evolution of SARS-CoV-2, a publication that can be downloaded for free on the NASEM website, where we talked about the need to sequence SARS-CoV-2. Again, a major contribution of Paul's to viral biology and society. The title of Paul's talk today is Phage Therapy to Combat Infections by Antibiotic-Resistant Bacteria. Paul, thank you for taking the time to be with us. We look forward to a great lecture.
Oh, thank you so much. That was a wonderful introduction. This is indeed, it's a huge honor for me to give this talk. Not only was I, as it was mentioned, a postdoc at NIH, but of course, my funding and my great appreciation for the NIH mission, is an obvious thing that would be on my mind. All right, I have a fair amount of ground to cover, so I'm going to get started. And I will begin with a punchline. So these 3 pictures might intrigue you. The one on the left is a classic approach where you can take natural samples and look within them for bacteriophages. These are the viruses that are able to infect bacteria. And the middle picture is a true image of one of the phages that we've isolated from nature. It had some properties that made it ideal to do what's shown on the right. This is a young woman who is in a hospital bed in Texas, and she has unfortunately suffering pulmonary failure due to a pan-drug resistant bacterial infection in her lungs. So a lot of what we're trying to do is to address the antibiotic resistance crisis. using an updated classic approach, and that would be phage therapy. So the same person on the left is shown again in the hospital room on the right. That's Ben Chan. He's a research scientist in my group, and he's literally showing this young woman a vial of phages that she's going to inhale over the course of multiple days to try and treat an infection that antibiotics cannot, as her doctor looks on. So I'll tell more of her story in a bit. I'll begin with this disclosure statement. I'm a co-founder of Felix Biotechnology. This is a company that seeks to commercially develop phages for use in human therapy. I'll show just 2 slides at the beginning here, a bit of the scope of the work that I do in my group, but I'm only talking about a very small subset of this today. As Julie mentioned, I have a broad interest in microbial evolution, and we study this in many ways. We look at the genetics, genomics, and evolution, especially of viruses, in the face of environmental challenges. And one could do this at very many different levels of selection. Shown on this slide is perhaps one way to think about virus emergence at different levels, whether they are very proximate interactions, such as binding of viruses to cells and how that can change over time, tissues, the movement of viruses between tissues of different types and the constraints that could exist there. Of course, human immunobiology, just like any host that is able to mount an immune response. This is something that we care about in terms of the spread of a potential virus. We're thinking about that so much these days. And the routes of transmission, there are very different ways that viruses and other microbial pathogens get around. So we take this into consideration when we do the research in my lab. And the last is, since I am an evolutionary biologist, I'll talk about that more in a moment, I have a great appreciation for biodiversity, including virus biodiversity, and the ecosystem level changes that can occur on this planet would affect all biological entities, even the smallest ones, viruses. The breadth of my research spans pretty much 3 compartments. On the left, I'm at heart a basic researcher, and I really enjoy questions that are at the leading edge of evolutionary biology. There is a long-standing tradition of using quantitative approaches, mathematical modeling, etc., to study evolution problems, and we do some of that, but we also do plenty of laboratory experiments. So on the left is just a couple of reminders of fitness landscapes, as well as trajectories of allele changes in virus populations, which we can look at intensively in the laboratory. In the middle is more of this ground that could be called ecology and evolution of infectious disease. Here we would be more interested in looking at the evolution of pathogens themselves. For example, we do experiments on dengue virus evolution in tissue culture and even look at the performance of different dengue virus variants in mosquitoes that we rear in the laboratory. But on the right is more of what I'm going to talk about today. And this is the fact that you can apply evolution thinking to disease and health problems. People call this evolutionary medicine. We do plenty of this. And what I'll talk about today is the application of evolution thinking to try and address problems that are not so easily addressed anymore in biomedicine. And that would be the rise of antibiotic resistance. The presentation objectives are listed here. I essentially am expecting that you'll learn something about natural selection and how it can often produce trade-offs. This is a very key aspect of evolutionary biology that we're capitalizing on when we do our phage therapy work. You will learn that phage therapy is indeed a classic approach to treat bacterial disease, but this method has some benefits as well as costs, and we're trying to update it so that the costs are more minimal compared to the benefits. And also, I hope that you will understand that clinical data show great promise for phage therapy. We have used it successfully to treat patients on an emergency basis. We're getting ready to do a clinical trial, and yet my talk will remind you that there's a ton of basic research that needs to happen as we move forward. So to begin, the antibiotic resistance crisis. This could be summed up in many ways. I'll be brief because I think this audience pretty much is familiar with the problem. There's an increasing proportion of bacteria that have resistance to commonly used and previously approved antibiotics. This is a global problem. It's threatening lives all over the world. And this projection map in the upper right is rather sobering because by 2050, it is expected that this would be a leading cause of death in the human population. Now, on the lower left, unfortunately, the problem could be described in many ways. Why don't we have as many antibiotics as we did in the past? Well, one practical matter is there's very little money in this. If Big Pharma is producing antibiotics that are destined to pretty much be placed on a shelf as last resort drugs, then they don't get the return on their investment. And they have to keep the doors open to their businesses. So this is understandable. And yet, if you rely on the halls of academia and NIH and other places to do antibiotic discovery, this is still possible. Nevertheless, there has been what's been called a discovery void recently. So we are pretty much running out of new antibiotics, and the problem is that the old ones don't work well. So let's turn to this old idea of phage therapy. Lytic phages have a particular kind of a reproductive cycle shown in the cartoon on the lower left. If a phage encounters a susceptible bacterial cell, then it can infect and take over the machinery of that cell and its metabolism to turn it into a virus production factory. It can then have these virus particles exit through a process that destroys the cell. That's called lysis. Now, long ago, this was seen in by the early discoverers of phages, including Felix Durrell. He was a co-discoverer in 1917. He had only one faculty appointment in his career. It happened to be at Yale for a few years. But early in his career, he was very instrumental in helping to found and do the early work at the Eliava Institute, which still operates today in Tbilisi, Georgia. That is the country, Georgia. So this is a long-standing tradition in the USSR and more recently Russia, as well as Poland and other Eastern Bloc nations. They continue to do work on phage therapy, and they started doing this before antibiotics were accidentally discovered by Fleming. The point here is just to remind that this is old technology, but I'll talk about the limitations to it. So the benefits could be obvious. You have specificity of a drug, if you would consider the phage a drug, potentially. It has specificity to its target, and that's attractive. It also is a drug that self-amplifies, and that's very unique. So this is a drug that you could use with minimal dosing, and it will make copies of itself, as long as it has susceptible cells to infect. Now, there are some costs that are somewhat related to those benefits. One is that if you have too much specificity, that gives you limited spectrum. And a drug with too limited of a spectrum, of course, is not going to be that useful. There could be a difficulty in delivering it to the infection site, depending on what that is in the human body. There could be immune recognition over time. You undoubtedly will eat, drink, or simply breathe in phage particles today, and in general, you don't mount a big immune response to them. We, like other species, are essentially in an ocean full of phages because these are the most plentiful things on the planet. And yet over time, if I'm using phages at a high level and putting them in the human body on purpose, then the immune system may catch on. And the concern is that you can't use the same phage on the same person more than once. But as an evolutionary biologist, I'm very keen to examine this problem and see it through the lens of the evolution of phage resistance. There are many defenses that bacteria have that are so-called innate, but they could change over time through evolution, as well as more or less acquired. And those are shown on the left and the right in this cartoon. On the left, things like restriction enzymes will cut up recognized phage DNA when it enters the cell, and we've capitalized on restriction enzymes by using them in the laboratory a lot in recombinant technology, but this is something that evolved so that bacteria can defend themselves against incoming genetic elements like phages. There are other mechanisms that I won't go into, but I will mention on the right, a more recent discovery than the ones on the left would be CRISPRs, where this is a more elegant system where over time there are cassettes of information that are built up in the bacterial cell to be able to immediately recognize some foreign invaders, especially plasmids and phages. Now these can build up over time. It does take time, and those cassettes have a limited amount of genetic information that they can carry, but this is a relatively recent discovery that is amazing that it kind of missed our eyes in microbiology for some period of time. So the point of the last slide is that it's a reminder there are many ways that bacteria can evolve resistance to phage attack. So how is it that we think that we could be successful in our efforts? Well, in my career, I've studied a lot of what are called evolutionary trade-offs. And this is the fact that evolution often involves compromises. If natural selection is improving a trait, in other words, taking the population to a higher fitness within its environment, it doesn't mean that other traits are also improving in fitness. There could be kind of performance liabilities as you evolve traits. And I'll get into that and how it works for us in phage therapy to our advantage. But first, an example from human biology is that what do we do that is different and perhaps better physically than other animals, including our close relatives? We can hurl objects with accuracy and speed. And you see this in professional baseball players and softball players. This is quite different than the abilities of our nearest relative, the chimp. And here you see a chimp, a young chimp, is able to hang from a tree branch by one arm for an extended period of time. And that's something that we cannot easily do. So the fact is we have these closely related species and they're quite different in their abilities. But if you just look at humans and their traits, the fact that we're the only great ape that walks around on two legs all of the time gives us vulnerability to neck and to lower back pain. So this is just a reminder how evolution can take you down a path, but some traits will improve and others can lead to liabilities. And that's exactly where I want to go with phage therapy. The innovation that we had is to find phages in the classic way in nature, but to find them and especially understand their mechanisms of binding for a particular purpose. And that's shown in this cartoon. If you consider two phages here, hypothetically, this is a bacterial cell, and you'll see that these orange and red things here decorating the cell, these are efflux pumps. These are complexes of proteins that pump antibiotics out if they get into the cell. And this allows some bacterial pathogens to be broadly resistant to antibiotics. Hypothetically, if you find this phage in red, it could be binding to this cell by attaching to the outermost protein of an efflux pump. And if it does this, the phage is presenting a problem to the bacterial population. It will kill it because it's a lytic phage. It will also select for the bacteria to change to overcome this problem. And in doing so, they may alter or even forego those efflux pumps as the binding sites for the phage. Of course, this is going to make them more sensitive to antibiotics, and that's very attractive. A faster example would be this purple surrounding the hypothetical bacterial cell could be a capsule that is shielding it from detection by the human immune system. And this would constitute a virulence factor. If you find a phage that is specific to that capsule, then it will select for the bacterial population to overcome the problem by perhaps altering or even removing the capsule, making it less virulent. So what we assumed is that we could go out into nature and find phages that will naturally bind to the myriad types of structures and mechanisms that different bacteria use as virulence factors and antibiotic resistance mechanisms. Some of them are shown here. So this was an assumption, but a big take-home message from my talk is that the assumption was verified and we were able to do this. It's pretty easy to isolate phages because you can find them everywhere. You can look in pristine environments. You could look in heavily altered environments. Humans have, for example, contaminated. You can look in wastewater treatment plants. You can look on farms of domesticated animals. So phage hunting itself is pretty easy and affordable to do. Once you obtain phages, such as a library that could be useful against a target pathogen, you want to find out more information about these phages, and that's what we do. And here, I think phage genomics is really crucial to phage therapy. You want to know what you're dealing with. Way back in the day, in the early 1900s, of course, there was a poor conception of even what genetics was and how genes and hereditary material moves across generations. Now we have an ease of obtaining genetic and genomic data. So here is a very busy slide that's basically taking roughly 100 phages and looking at their relatedness to one another and placing them into different virus families. And they are all specific for infecting Pseudomonas aeruginosa. We can do things like examine these phages genomically to make sure that they don't have toxin genes or other things that would be dangerous if we were going to groom these phages for use in therapy. So there's a variety of things, variety of information we can get from the genomics. Now, a concern in phage therapy, a longstanding concern, is that phages are too specific to be useful. And I mentioned that early on. And I would assert that that is pretty much an assumption based on some well-studied phages of the last century. And instead, our work and others shows that if you go and find phages naturally and bring them into the laboratory and challenge them to infect different strains of a target bacterium, you can find some phages with very good genotype coverage of that bacterium. So this unpublished data set led by Ben Chan and my group, here we have on the left some candidate phages, and this is a matrix where the dark squares are indicating where that phage on the left is able to infect a clinical strain of Pseudomonas aeruginosa that we obtained from the CF Foundation. And above all of that is a relatedness of these bacteria based on their genomics. So what we try to do is make sense of this matrix in terms of the relatedness of the bacteria. But an immediate thing that you can see is that there's a lot of dark blue in this matrix. There are a lot of phages that are good at infecting multiple strains of bacteria. Which might you use for therapy? That's kind of a golden question. Is it better to bundle them together and use cocktails of specialist phages? Or is it better to focus on trying to find one that is able to cover as much genotype space as possible? And maybe you'll find multiples of those and you'll bundle together generalist phages. I would say that this is an open question, and people are moving forward, including us, with their favorite approaches. But I think we need to have more of a general understanding of the best approach to go forward. And that's part of what we do in my lab. Now, here is a version of that data set as we step away from it. And I know you can't make out the details, but it is to impress upon you the size of the data sets that we grapple with. So if you have this giant matrix and it's giving us a lot of information phenotypically about what the phages can do, as well as genetically and genomically, how do we sort through this to make sense of it in a future world where a physician might have the genotype of an infecting strain and they would like to know immediately from a phage library what is the phage that would be useful? Well, we're trying to take machine learning and artificial intelligence approaches to this problem to see if we can figure this out. If you are wanting to use a phage in therapy and drive a useful trade-off like I talked about, you need to know what the phage is binding to. And unfortunately in virology, this is a tricky thing. It's not that simple to figure out what viruses bind to on the cell surface, and that is certainly the case for phages. We had a paper published last year that is similar to what a lot of other groups are doing, and we are trying to use high throughput discovery of cellular receptors that phages use in binding. And what we did is take an existing technology, transpose on mutant libraries created through NSeq. And the cartoon on the left kind of walks you through the approach. You can create your variable library of the bacteria, you can subject them to phage selection, and allow the bacteria to grow out. And what that will do is enrich for the phage-resistant mutants, those that have genes that are knocked out that are important for the phages to grow, and therefore you find the outgrowth of the bacteria that are resistant because those genes and their proteins are missing. So here in the lower left is a cartoon where I'm keying you in on the blue here. This is the enrichment that we would like to see in these analyses for the genes that would be the statistically relevant and hits in our screens. So this works very well, for example, in a control. Phage T6 is a very well-characterized phage of E. coli. And in the screen, we were able to find that TSX porin is the thing that gave us the greatest statistical significance. This has been known for a long time that the TSX porin is what this phage uses for binding and entry into the cell. On the right is a novel phage from nature that pretty much all we knew about it is that it was lytic and it could kill E. coli cells. It also happens through our screen that it is found to bind to TSX. Now that's just a coincidence, perhaps, but we do talk about this phenomenon in the paper where we are documenting the binding characteristics of multiple newly discovered phages. And the question is whether there are some kind of constraints on what phages can bind to on the cell surface versus the possibility that they can bind to just about anything if they evolve the ability to do so. So another cartoon from a recent review, this was in Current Biology last year. This says again what I mentioned earlier in the talk, and that we are trying to find phages that interact with bacteria in a particular way so that they can drive these favorable trade-offs that work to our advantage. And these two examples here on the left, I actually already covered those, efflux pumps and this capsule around a bacterium. Now I want to say something briefly about lipopolysaccharide or LPS. This is structurally very prevalent around the bacterial cell, and sometimes phages use this as a receptor to gain entry. And the point is that the depth of LPS can be important for the ability of that cell to restrict the entry of antibiotics. So if you consider phage selection, it can trim down that LPS to a shallower level, and this could allow more easy entry of antibiotics. So this is yet one more way that we could drive a trade-off. So I'll go through in the next few slides some published material. I'll do it pretty quickly because you can go look at this paper that was published last year if you want. This is a great example in a model system of E. coli where this phage that we discovered in nature, and we've characterized it to show that it is very dependent on receptors on the cell surface, and that this can drive the evolution of antibiotic sensitivity in two ways. So this phage U136B, it is interacting with this Toll-C efflux pump of E. coli. Here's a structural side view of it, where the Toll-C protein is the outermost protein of this pump. So there's lots of functions for efflux pumps as listed here, but in the middle, I'll turn your attention to knockout challenge strains of E. coli and the ability for these phages to grow or not on the strains. And clearly, the best growth is happening on a variety of these bacteria, regardless of knockouts, except for this Toll-C knockout. So when that protein is missing from the cell exterior, these phages have a big problem and they can't enter. That gives us confidence that Toll-C is indeed a receptor. But here on the right is the trade-off. So if you consider the growth of wild-type E. coli, an engineered Toll-C knockout strain, and a spontaneous phage-resistant mutant that is resistant to this phage that I just described, they all grow equally in the absence of tetracycline, but in the presence of tetracycline, the Toll-C knockout and the phage-resistant mutant cannot grow. And that's what we would expect because of the low function or the absence of the Toll-C efflux pump. But the plot thickens for this phage because it's binding to something more than efflux pumps. It actually is binding to the mid portion of LPS. This is a side view of LPS with the outer portion of the cell to the top here. And in the middle here, in the colored rectangles, this is the portion where this phage is now known to bind, judging by these different knockout challenges of bacteria shown here in the middle and the inability of the phage to bind on some key knockouts. But the consequence of the bacteria evolving phage resistance by changing LPS instead of Toll-C is shown on the right. Here, for these various strains of the mutants that we isolated in our laboratory, they now show a lesser ability to be resistant to colistin, a different antibiotic than tetracycline. Now, taken together, what this means is that we have evidence of two different pathways by which these bacteria could evolve phage resistance, but it leads to this problem of a trade-off. Okay, here's the bad news for what we are attempting to do and others in phage therapy or any system where you're predicting evolution. There could be genetic solutions that don't give you what you hope to see. So we described in this paper how both collections of mutants that have changed efflux pumps in response to phage selection, as well as those that have changed LPS, you can look at their resistance versus sensitivity to an antibiotic relative to that of the wild-type bacteria. And here, the wild-type E. coli have a certain minimum inhibitory concentration of tetracycline that they see. And this bar here on the left is what we would hope to see, is that mutations for phage resistance lead to greater sensitivity. But that's not all you get. You have actually this bar here with a fair number of the mutants, almost half of them, give both phage resistance as well as greater resistance to the antibiotic. So the LPS here on the right-hand graph, that gives us a better overall prediction that matches what we would hope to see. And yet, what you have to think about is if you deploy this in a patient and evolution happens next, which of these mutational paths would be possible for the bacteria? That depends on the competitive nature of the mutants. How well will they outcompete the wild type, and how well will they outcompete other mutants? In the laboratory, if we grow the wild type bacteria here on an automated spectrophotometer readout over time, the solid line is the wild type bacteria, and the Toll-C mutants have growth that is almost indistinguishable from the wild type. So they have very low fitness cost to these mutations in terms of growth, whereas the LPS mutants have debilitated or lower fitness growth. By this, we would expect that Toll-C mutants are going to be the ones that are favored in competition over time. If they are the ones spontaneously occurring and solving the problem of phage resistance, they'll probably outcompete those that have LPS changes. And indeed, that's what we saw in the laboratory when we do coevolution experiments. First, look at the red lines here. These are multiple lineages of the bacteria evolved in the laboratory over time in the absence of phage. But we can track in those bacterial populations, do they change at all in their levels of tetracycline resistance? We don't expect them to because there is no phage selection. So it bounces up and down, but it pretty much stays at the wild type level. Whereas in the presence of phage, when we're letting coevolution occur, here the bacteria need to solve the phage problem. And you'll see that over time, there tends to be this drifting downwards of the tetracycline resistance phenotype. And that is through data that I'm not presenting here, that we show in the paper. And we have observed that the TOL-C mutations are responsible more so than the LPS mutations, because the TOL-C mutants have competitive superiority, and the consequence is that that can lead to reduced tetracycline resistance. So I'll take 2 more slides to talk about a different way of using a phage, but now we're trying to toggle virulence rather than antibiotic resistance before I dive into some clinical data. So this is yet one more phage that's new to science. It's called TIVP-H6, and that stands for type 4 pilus H6. This binds to type 4 pili of P. aeruginosa, and it selects for phage resistance that reduces piocyanin production. And you might ask, well, why is that? That's due to a gene network that links up not only the production of the type 4 pilus, but also the production of piocyanin in these bacteria. Now, piocyanin is a very dangerous virulence factor. It has toxic effects, including inflammation and oxidative stress in human cells. So the pilus loss that these bacteria tend to undergo to solve the phage problem, this can also reduce their ability to form biofilms and to move about through twitching motility. In the laboratory here is a very characteristic blue-green color when you grow bacteria up that are able to produce this piocyanin that has a characteristic pigment to it, whereas if they cannot produce piocyanin, they grow more like what you see in the test tube on the right. They don't make that chemical, and therefore it does not change the color in the test tube. So what we have observed in this unpublished data set, and it's a little busy, but I'll walk you through it, is it's showing you the relative piocyanin production when you put P. aeruginosa into different treatments. If you put them in the presence of Cipro or tetracycline or some other antibiotics for which they are resistant to it, it actually can drive up their production of piocyanin, and that's certainly not what you would want to have happen in a patient. Erythromycin is sometimes given to people to calm down this inflammation problem of piocyanin production, but it doesn't mean that it would be killing the bacteria. Instead, it just calms the inflammation response. Here on the right, the final bar here in this data set is a phage that we discovered that does both. It kills the bacteria, but it is also selecting for the bacterial population to solve the problem by reducing piocyanin production because they are reducing or changing their type 4 pilus. So all in all, what we have here is good evidence that H6 is a great phage to do this and can work well in combination with erythromycin. It can even work well in combination with Cipro because you'll see that the deleterious effects of Cipro go away in that circumstance. All right, so before I turn to the clinical data, I have to remind you of, once again, we are seeking to do trade-offs and to have bacteria selected to do things that are less biomedically concerning when we deploy phage therapy. But there are things that we cannot control, but we need to examine in the laboratory prior to using some phage because it could give you, on average, the opposite result, a trade-up. I mentioned trade-ups a moment ago, but if you consider this final example in this cartoon, imagine you have a phage and an antibiotic that are entering the cell through exactly the same porin. In this case, if the bacteria evolve resistance to the phage, they're also going to evolve resistance to an antibiotic, which is clearly not something that we want. And we have a paper that is in the process of revision that shows if you look at phage T6, classically known to enter through the TSX porin, as well as U115, I show you the data for that novel phage earlier, they both increase bacterial resistance to albacidin, which is an antibiotic that enters cells in exactly the same way through the TSX porin. So if you look at the proportion of mutants in a collection that we obtained in the laboratory, you find that across the TSX gene at different locations, that there are a variety of mutational ways for either phage T6, phage U115, or albacidin to lead to the evolution of cross-resistance to all three. So it doesn't matter what you throw at the bacteria, they're able to solve the problem. And unfortunately, they solve it in a way that they're able to restrict the entry of a phage that they've never seen before. And they can also be resistant to an antibiotic they've never seen before. So this is a lesson or a cautionary tale that as you move forward, you want to know what you're doing in phage therapy because mechanistically things could turn on you. So the last part of my talk concerns clinical evidence, and I'll go through this largely unpublished data. I'll touch on a little bit of published data, but now I can show you, how is this actually working in emergency therapy? So what we have is nicknamed as a pond to bedside pipeline. And I showed you this earlier. It consists of phage discovery, characterization, and ultimately treatment. So we're not the only ones doing this. There are other research groups that have been successful with this, especially emergency patient therapy. There are companies that are trying to harness this in a commercial way and moving forward. And there's even a book written about phages, lytic phages, and their ability to destroy bacterial cells as related to a well-known patient case that happened at UC San Diego. Now, our first case is published. I'll only mention it quickly, but it was quite a success story. And it was, since it was our first success, I'll highlight it here. We got US FDA approval to treat a chronic biofilm infection in a man who had undergone an aortic arch replacement, which is not a trivial surgery, but of course it's a surgery that happens frequently. Unfortunately, this man had Pseudomonas aeruginosa that infested the aortic arch, and it was drug resistant. So they couldn't treat it with antibiotics, and he was seen as too elderly to have that infested piece of material taken out through surgery and to go through the surgery again. Instead, he volunteered for experimental emergency phage therapy, and we got the FDA approval to do this. So we placed in his body a phage plus an ordinarily useless antibiotic that we predicted would synergize with the phage, because the phage was going to kill the susceptible bacteria and then create a bacterial population that was sensitive to this antibiotic and make it useful again. That's exactly what happened. And this is a picture of Ali when he returned to work here in New Haven as an ophthalmologist in his later years. So he had quite high quality of life. In the end of his life, he ultimately did succumb to heart failure, but he was able to go back to work. And he and his family were super appreciative of how we helped him. In Western medicine, and I'm saying that purposely because Western medicine has turned away from phage therapy basically in earnest around the time of the Cold War. But now Western physicians are taking note of whether phage therapy can help deal with the antibiotic resistance crisis. So if you consider magazines on their popular press and TV, they've gone on to this and they've tried to examine whether phage therapy is something that's here to stay in Western medicine. So one more case, this is an unpublished case, but I'll go through it again quickly. This is the young woman who was in that hospital room that I showed you earlier. So in her case, she was undergoing many years of lung failure, and she was pretty much headed for a lung transplant at best. Pre-phage therapy, they had found out about our work with the prior patient and contacted us because of this alarming outcome. So she was undergoing pulmonary failure, and the bacteria that had infected her lungs were pan-drug resistant to all of these listed antibiotics here on the left. So she had very poor lung function. Her FEV1 values were very poor, poor ability to exhale air from the lungs in 1 second. Her exercise performance was poor, and she had very high density of these bacteria indicated in her sputum. And as I said, they were pan-drug resistant. So you can see in green here the long years of decline for this young woman. And when we administered the therapy, her lung function improved very quickly and it trended upwards. So these are statistically different regressions. On the left, it is decreasing significantly. On the right, it is increasing significantly. But here's the better outcome. After phage therapy, she had improved lung function. She had better exercise performance. We did not eradicate all of the bacteria that were infecting her lungs, but it pushed it down to a very low level. And best of all, they were now largely sensitive to antibiotics. So her doctor now has many options in treating her if she has an exacerbation. As a cystic fibrosis patient, it's almost inevitable that she will have exacerbations from time to time, including this outgrowth again of the bacteria in her lungs. But now it is possible to treat them with antibiotics. So she is one example of many people who have a problem summarized here on the left. If you are born with CF, if you develop bronchiectasis, if you have chronic obstructive pulmonary disease, you have a lung structure that unfortunately makes you quite susceptible to a chronic infection caused by increasingly antibiotic-resistant bacteria. So I talked about these first two cases at the top of this table. Filling out the rest of them, you can see these are mostly CF individuals or bronchiectasis. Mostly they are having multi-drug or pan-drug problems with Pseudomonas aeruginosa, but in all cases, we were able to treat them successfully with emergency phage therapy. What we're trying to do is get together their cases into a bundled case report, and the data goes something like this. These are unpublished data, but the way we deliver the phages is through nebulizer. So they inhale in the phages over the course of multiple days, and we can take from these patients sputum samples that they cough up. And from that, we can examine many things. Not only are we looking at their lung function, and this data set shows you how it improves over time on average, but we're also looking at the bacterial load in their sputum for the target bacteria, and that drops, and that's certainly going in the direction that we want. Here on the lower right, we often see that the antibiotic resistance flips to sensitivity, as predicted. The inflammatory molecule reduction, which is also favorable, I have to explain a bit more of why we obtained that result. So what we did was these longitudinal analyses of patient sputum samples, and if you remember the phage I talked about earlier that targets type 4 pilus, what you expect to see if you take clones from sputum is at the beginning here, they're mostly able to grow in the laboratory and they produce piocyanin with that bright green color. But over time, they should not, if our therapy is working in the patient the same way that it's working in the laboratory through in vitro studies. Here, as we see Pseudomonas aeruginosa clones from a single patient taken over time, different time points, this piocyanin production and the concentration in the media troughs out, as we would expect. And the observed piocyanin reduction is statistically significant. Working with this, unfortunately, I'm forgetting to sometimes mention the folks who were the primary movers of the research, but you did see their names and pictures along the way. I'm now remembering to say that John Koff and Gail Stanley, they are two MDs who are working with us on this problem, and they helped generate these data, which are further confirmation that if you take the clones from a treated patient pre, during, and post-therapy with phages, and you look at these markers, you can find the favorable outcome that you expect. Essentially, we grow the clones out from their sputum, and then we take the supernatant from those bacterial cultures and challenge airway epithelial cells in tissue culture to look at how stressed they are and their inflammatory markers. And we find that things go in the right direction that we would predict. Now, what we are doing is clearly evolutionary prediction, and admittedly, it is in a black box because much more is happening in the patient than is happening in vitro. And yet things are going well. In fact, I would say they're going remarkably well, given that we are largely working in a black box. Now, the pandemic has me thinking about many things like you, and my work actually does involve virus emergence. So I'm now going to relate a little bit of what we are challenged with in virus emergence and predicting it to how this also relates to what we want to see in viral therapy and predict it. So the goal in emergence is to better predict the potential for a virus to emerge on a new host, especially humans. And we would like to know, why are some viruses good at infecting multiple hosts? We have certainly experienced coronaviruses and their emergence in the pandemic. We've seen HIV emergence. We've seen certain strains of influenza virus emerge historically in humans. And the overarching question that we've looked at in my research is whether generalist viruses, those that are sort of groomed through selection and evolution on multiple hosts, are they more likely to emerge on new hosts in the future? That's just one line of questioning that we pursue. But now I'm going to try and relate this to what I've talked about for phage therapy. One goal is that we want to better predict which phage have the potential to broadly kill the target bacteria. And we would like to know, why are some phages good at infecting multiple bacterial strains? These make them good candidates. However, there's something that we don't want to happen. We don't want these generalist phages to easily emerge or grow on commensal strains of the microbiome. We would be then recreating problems that are associated with broad spectrum antibiotics, things that devastate the microbiome, and then you have to be concerned about these off-target effects. So I would say that hand in hand, some of these questions can go together. I'm excited to talk about this a little bit more tomorrow at another talk at NIH for the Lambda Lunch. So in our evolutionary prediction, we have to think of phage therapy affecting things other than what we are directly predicting. It could be interactions with your cells. There could be interactions with other phages within your body. There could be changes in bacterial traits aside from the ones that we're predicting. There could be non-target bacterial pathogens that for example, might have a competitive advantage. If you push one pathogen out with phage therapy, what's the consequence of other pathogens in the human body being able to successfully compete better for infection? And other non-target pathogens that are not even bacteria, these could be viruses of eukaryotes and fungi in the human body that could also be indirectly benefited by you introducing a phage purposefully to kill off the pathogen that's there. And just in general, what are these effects on the resident microbiome? So I clearly don't have enough time to go into all of that, but just a little teaser, some unpublished data with Jennifer Bomberger's group at University of Pittsburgh, Paula Zamora is a postdoc in her group, and they've confirmed some of what is seen in the literature, that if you do tests in the laboratory for the ability of phages to actually enter cells of mammalian origin and human origin, they can do so. So they can't replicate, but they can sometimes enter and even mount an immune response, the production of interferon. So we are, of course, interested in how that occurs with the phages that we are deploying in therapy. But there are even some traits of our phages that we kind of underestimated how they could benefit if deployed in therapy. And here on the right is a reminder for me to say that one of the phages that we've been using is a so-called jumbo phage. It is a large genome-sized phage, and it has a very interesting ability to enter a bacterial cell and shield away its reproduction, its replication within the cell, so that CRISPRs cannot detect it and attack it. So it sort of has a built-in anti-CRISPR defense. Of course, that might be useful if you deploy a phage that has some additional tricks in its arsenal to work in your favor. So what I talked about today is mostly Pseudomonas aeruginosa, and I didn't have time to mention the very many other bacterial targets that we are developing phage therapy against. We're doing the discovery in earnest. We're doing emergency treatment, what I'd call a preclinical phase. And now we want to move into a clinical phase. So I always kind of consider these trains running down parallel tracks, and some of them are further ahead, getting to the station. So the next steps include a new FDA-approved clinical trial that we have coming up at our hospital here in New Haven, at Yale New Haven Hospital. And we're going to test the ability for phages to protect prophylactically the lungs of healthy CF patients who are volunteers. So over a multi-month period, they will inhale phages, and we will track how well these phages prevent them from experiencing exacerbations relative to a placebo group. And it goes by, I guess it's called a crossover. I'm not quite remembering the true name for it, but I think that's the case, that all of the individuals who participate will get the placebo as well as the phages at some point in the clinical trial. They, of course, won't know it, but we can look at these individual volunteers to see how they do during the placebo versus the phage prophylaxis time points. We teach this technology to students at Yale. There's actually a very popular course across the country that was developed by Graham Hatfull at University of Pittsburgh and also supported through HHMI. But we teach our own flavor of this to Yale undergraduates, and this has been an awesome way to give students an entry into authentic research. And we also take the technology to places in the world where a need for more affordable drugs could be something of the near future. For example, we did a workshop in West Africa teaching physicians and researchers there about phage therapy, showing them how they are isolated and characterized. So it wasn't the idea of it immediately launching the therapy, it was just building their ability as researchers and physicians to understand the technology and to move forward. The last I'll say is that non-human systems are also affected by antibiotic resistance problems. There's plenty of antibiotic-resistant bacteria in plant and animal agriculture as well as aquaculture, and this is threatening the food supply. So there's an earnest need to address these problems there as well. Zoo and aquaria animals can suffer multi-drug resistant bacterial infections. We were doing the work that I talked about today, and some veterinarians at an aquarium in Florida contacted us because they had a sea turtle that had a chronic bacterial infection that was multi-drug resistant, and essentially the animal had stopped feeding and it was slowly in the process of dying. Not working on its particular bacterium, we still helped these individuals out and saved this animal's life by looking for phages that were specific to multi-drug resistant Citrobacter frundii bacteria. So I'll end with acknowledging the many folks who have done the work in my laboratory. I've benefited by the current and past folks in my research group are just terrific. They are very excited to do the kind of research I talked about today, as well as our very many partners here on the right, folks in my lab, as well as those that we collaborate with, showing them here. And since I'm running short on time, I won't kind of highlight them individually. I will say that the individual here, Deepak Narayan, he was the surgeon on the very first case that we did for the man who had an aortic arch replacement that was infested. So he believed in what we could do. He had run out of options for this patient, but he believed that this technology had some promise. And if he had not had that confidence in what we were doing, we would not be able to then move on and help individuals soon after then. So I always give credit to Deepak that unfortunately he left us way too soon. He died of cancer on Christmas in 2018. All right, so with that, Julie, should I stop sharing? Maybe, is that the better way to go?
Sure. First, thank you, Paul.
Thank you. I appreciate it.
That was a great lecture.
Thanks.
And I really appreciate your taking us from discovery to bedside and thinking about all of the ways in which we need to understand evolution and fitness landscapes to really achieve some substantive goals.
Oh, thank you.
You know, you mentioned it briefly, so there's a ton of questions in the chat box, but I'll just take the host prerogative and ask you mine. You mentioned the jumbo phage, which are a really interesting group because phages tend to be very compact genomes.
Exactly.
But there's been a recent discovery of some that are about 200 kb or well, that's the basic definition of a jumbo phage. So I guess I wonder, does that open the door, or even with the other phage that you're describing, are you considering to use phage natively, or are you also considering to engineer the phage to bring that in to sort of make sort of a second generation version? What are your thoughts on that?
That's a great question. So I have mixed feelings because there's mixed opportunities. So basically the US FDA has been very supportive of what we do, and the barriers are fewer because we're not using genetically engineered stuff. Okay, so there are ways that we could sort of leap over early phase levels of trials because phages are considered GRAS, generally regarded as safe by the FDA. So that helps us. And yet, you know, on the company side, people who are chasing this for intellectual property, there would be a greater interest in genetically engineering. And the question is, you know, is that an okay way to go? But what you asked about is really what one needs to consider. If there's not enough tricks in the bag of some phage when you're deploying it, and you really need to put something else in that would benefit the patient, then you really should go the engineering route. So essentially, we are looking at both for the patients who we've done emergency therapy so far, and for our upcoming clinical trial. These are unaltered phages. These are straight from nature.
Yeah. Okay. So I'll start with some questions about the clinical work, and so a lot of questions asking about opportunities here. And so I have a question from well, Chris will tell me later if I'm supposed to say who's asking this question, but I'm going to just kind of aggregate a few questions here about the opportunities. So for example, mycobacterial infections of the lung, TB or non-tuberculosis mycobacterium, and any comments on case studies of Mycobacterium abscessus infections where the dramatic results have been seen but are not durable with a rebound in disease in some cases. Like, how do you think this is all happening?
Yeah, so I mean, there's a great need in so many different bacterial systems for this kind of technology, or some technology. You know, this doesn't have to be. I should say that very clearly. I don't necessarily believe that this is the panacea that's going to, you know, solve everything. I think what's exciting right now in terms of the antibiotic resistance crisis, it's a little bit like, you know, the pandemic. You know, you want to have multiple vaccines to try and solve the problem. You don't want to just have all your money stacked up on one. So this is what is happening. There's a lot of attention. That's a bit of a deviation from what you asked me. But so the real trick is, how easy is it to find useful phages that would do this? And the story of non-tuberculosis mycobacterium is a wonderful one because there's a huge library of these phages that are part of that HHMI course that was developed by Graham Hatfield, but they took extensive searching for them to find a handful of phages that worked for a well-known case in the UK of a young woman who was dying from multi-drug resistant form of that bacterium. And they had to engineer it. They had to take out the genes in a couple of these phages that would cause them to have a more complicated reproductive cycle called lysogeny, where they basically go quiet within the bacteria and are inherited by daughter cells. That's not what you want your drug to do. You don't want it to occasionally go quiet on you. So I think the future is both bright, but also concerning. Why is it that we can't find phages for some targets, such as TB? Why is this? And it may be, on the one hand, that we just don't know how to culture the bacteria in the right way in the laboratory to make them present the things on their exterior, or we might find phages that exploit them. Or the other is that there's a real biological constraint. Phages are the most abundant thing on this planet, but it doesn't necessarily mean that all bacterial species are equally attacked by speciose phages. There could be actually some phages that are so good at attacking their bacteria that they outcompete other species of phages. And I think we underestimate that as a possibility. So I don't have a great answer for your question, but I think you can see that I'm trying to consider it holistically in terms of what are the possibilities as well as the constraints, so that people who are contacting me all the time about a particular system, how can we move forward? I want to give them the best advice.
Great. So let's continue that discussion. And I have a very specific question here about the woman suffering from cystic fibrosis who received the phage therapy. And the question is whether that created selection for different Pseudomonas aeruginosus strain that then was present or driving the infection, because the CF patients often have these mixed infections, and you may even have the Pseudomonas aeruginosa and other, like Staph aureus or other. And then how many phage, this is a very specific question, like how many phage are typically used in this phage therapeutic dose?
Yeah. So I'll take the second part first. We tend to use, I think it's about 10 to the 9 phages delivered in a dose in a nebulizer, and you're not going to get all of those making it through the nebulizer into the lung. You're going to get some loss, but it's pretty good. And then the other question then was with this. Wait, say it again.
Like about strain selection, are you going to basically just move the population from one strain to another?
Big concern, especially as you move forward with making this a general prophylactic, for example. So what the audience should understand is the vast majority of what we've been doing so far, these are people who are very desperate. I mean, they are in the process of lung failure. They're basically in the process of dying. So basically a downstream consequence of what you said is very true. It is known in the CF lung that P. aeruginosa would outcompete Staph aureus, especially in certain age groups of those individuals. So that was a concern. We are deploying a phage. We're trying to knock out Pseudomonas aeruginosa, at least a susceptible one, with this clearer path for Staph aureus to have what's called release from competition, right? And that didn't happen, but we were looking for it, watching for it like a hawk. And maybe, but I don't really have a mechanistic explanation, but I'll, wax philosophical a little bit. We couldn't eradicate the Pseudomonas aeruginosa anyway. We changed it so that it was a more favorable variant for biomedicine in case of an exacerbation. So maybe that's a better way to go. This is talked about a lot in the microbiome. Do you really want to take a scalpel in and remove? But in cross-community ecology, that's an open question. Is it better to remove something? Am I going to genetically engineer through CRISPRs and introduce them in mosquitoes to completely remove them? Or do I just want to make them incapable of transmitting malaria? And I at least believe that it's probably better to not completely mess with the ecosystem, but in some way alter it to your advantage. And I wonder if we sort of stumbled into that space. I believe that you might have asked me something else there, but I might have missed it. Is that kind of what you asked?
I mean, you took it in an interesting direction. And I think that was the question of sort of, and it's even what we talked about with antibiotics. It's like, do you want these general, come in with the big,
Sure.
sledgehammer that you can use it for everything, or do you want a very precise, that you have to then create? I guess that's the thing. Do you think we'll move towards having very personalized, where if you have this, if you have this microbe and this strain, and then we go into the freezer and there's going to be, these banks of them, and we think, oh, we'll give you a little of this, a little of that. And
so of course it's impractical to completely rely on personalized medicine. What we're doing right now, the irony is that that is what we're doing, because that is what we're allowed to do. We have not done clinical trials for general safety and efficacy yet in the phage realm. These are happening, including under our direction. So it's a little unclear. And but think about broad spectrum antibiotics. How many of them would actually be approved now? If they had to go through an approval process from what we know with the microbiome. So I kind of see it as it's a desperation moment, but then quickly we have to think, what is the general way that we could do this and have it be placed into people safely? Now, to be brutally honest, in some countries it's already been long approved, and you can go to the pharmacy and buy whatever suspensions of phages you want without a prescription. So it's a different space.
Antibiotics would still be approved because they're so incredibly effective. That's what we're seeing here, and that's the promise that you've showed us with your talk, is these, a new arm of biomedical infectious disease research that is very effective, but is also being developed with a lot of science and caution for the long-term benefit of society and these patients.
And I like what you said about the research bit. At the heart, I'm a basic researcher. I'm so I feel all this reward, and just so rewarding to have this go down this path. But so many more questions are created as you move forward with new technology, right?
Yeah.
So it's this, I see so many people coming into the phage biology field and biotechnology of phages taking off like a rocket, and that's really cool. That was not the case 20 years ago when I was cutting my teeth. Actually, I got that 25 years ago or more when I was cutting my teeth on phage research. So it's a pretty cool moment for phage biology.
Well, with that, I see we're at the top of the hour, so I'm gonna again thank you for presenting the Rolla Dyer Lecture, and this plaque will be sent to you in appreciation. And I only wish that if you went to your front door, there would also be home-baked cookies. But I know that people are looking forward to talking more with you, and I really appreciate you visiting NIH today. So
thank you so much
for your great research and talk today.
Well, I appreciate it. It's an immense pleasure. Thank you so much for the invitation.